Assalam Alaikum students. Welcome to the Bright Future Academy. Today we will study about the structure of proteins. Before studying the structure of proteins, first of all, you must have to know about what are proteins and how they perform their function in our body. Proteins are the most abundant organic compounds to be found in cells and comprise over 50% of their total dry weight. They are present in all types of cells and in all parts of the cell. Proteins perform many functions. They build many structures of the cell. All enzymes are proteins, and in this way they control the whole metabolism of the cell. For example, like hormones, proteins regulate metabolic processes. Some other proteins, for example hemoglobin, work as carriers and transport specific substances such as oxygen, lipids, metalloids, etc. Some other proteins, which is known as antibodies, defend the body against pathogens. So, proteins perform many functions in our body. And if you want lecture on any topic, you can comment me in comment section. Protein has specific properties which are determined by the number and specific sequence of amino acids in a molecule. Because each protein has specific properties depending upon the number and sequencing of amino acids and upon the shape which the molecule assumes as the chain folds into its final compact form. There are 4 levels of organizations which are described below, such as primary structure, secondary structure, tertiary structure, and quaternary structure. The properties of proteins depending upon the number and sequencing of amino acids, because there are 20 known types of amino acids. The first one is primary structure of proteins. F. Sanger was the first scientist who determined the sequence of amino acids in a protein molecule. After 10 years of his work, he concluded that insulin is composed of 51 amino acids in 2 chains. Insulin is a small protein which is used to maintain the glucose concentration in blood. One of the chains had 21 amino acids and the other had 30 amino acids, and total it contained 51 amino acids. They were held together by disulfide bridges. These 2 chains connected to each other by making a disulfide bridges between them. Hemoglobin is composed of 4 chains, 2 alpha and 2 beta chains. Another example of primary structure is hemoglobin. Hemoglobin is also a oxygen carrying protein which contains iron in itself. Each alpha chain contains 141 amino acids while each beta chain contains 146 amino acids. There are certain numbers and certain positions for the amino acids every structure of protein. Either it may be primary structure, secondary structure, tertiary, or quaternary structure. So the properties of a protein depending upon the sequencing and positioning of amino acids in it. Hemoglobin contains red blood cells, and hemoglobin basically carrying oxygen with it and transport this oxygen to the animal's body's tissues. Alpha chain and beta chain. Alpha chain contains 141 amino acids and each beta chain contains 146 amino acids in it. Hemoglobin also have a heme group, and in this way it is known as oxygen carrying protein because it binds oxygen molecule with it and transport to the body's tissues. As shown in another diagram, polypeptide chain in keratin and in hemoglobin globular protein are held together to form functional proteins. Globular protein subunit and heme unit are heme group. Here's the hemoglobin globular protein. Hemoglobin is a globular protein and keratin fiber is a fibrous protein because keratin is composed of various fibers. There are over 10,000 proteins in human body which are composed of unique and specific arrangements of 20 types of amino acids, which are known as essential and non-essential amino acids. The sequence is determined by the order of nucleotides in the DNA. DNA is also made up of some kinds of proteins such as histone proteins. Histone protein is present on the DNA which carrying a positive charge on it, and in this way DNA as a whole comprises of negative charge on it, and positive and negative charge both attracts each other and this bond is formed. The best example is the sickle cell hemoglobin of human beings. Sickle cell is a blood disease in which the red blood cells not remained in their original shape and change their shape from biconcave to another flattened shape. In this case, only one amino acid in each beta chain out of the 574 amino acids don't occupy the normal place in the proteins. In fact, this particular amino acid is replaced by some other amino acid. Sickle cell hemoglobin or sickle cell anemia is a blood disease which is caused when one amino acid out of the 574 amino acids don't occupy the normal place or either replaced by any other amino acid. The hemoglobin fails to carry any oxygen, hence leading to death of the patient. Because if hemoglobin fails to carry any oxygen, the oxygen would not be transported to the body's tissues and then the patient may be died. Here is the secondary structure of protein. Secondary structure of protein is much complex than the primary structure of protein. The polypeptide chains in a protein molecule usually don't lie flat. Each structure of protein has specific properties depending upon the sequencing and coding of amino acid. Because amino acids are the building blocks of proteins and proteins are the polymers of amino acids. When hundreds of amino acids of long chains combine each other, then a molecule of protein is formed. So the secondary structure is much complex than the primary structure of protein. They usually coil into helix or into some other regular configuration. The primary structure of protein lies flat, but secondary structure of protein coil into helix or into some other regular form. One of the common secondary structures is the alpha helix. Alpha helix like this, spiral form, spiral shape. It involves a spiral formation of the basic polypeptide chain. The alpha helix is very uniform geometric structure with 3.6 amino acids in each turn of the helix. Like starting from here to each one turn of this helix, 3.6 amino acid lies in each turn. Then again 3.6 amino acids lies and then this helix again turned. The helical structure is kept by the formation of hydrogen bonds among amino acid molecules in successive turns of the spiral. Beta-pleated sheet is formed by folding back of the polypeptide. Two important and main things in the secondary structure of protein. The first one is alpha helix and the second one is beta-pleated sheet. When these helix and this beta-pleated sheet is formed, or coiled or spiraled itself into some regular configuration, then the secondary structure of protein is formed. Then the tertiary structure of protein, and tertiary structure of protein definitely much complex than the primary and secondary structure of protein. Usually a polypeptide chain bends and folds upon itself, forming a globular shape. This is the protein's tertiary conformation. In tertiary structure, the protein will be in a globular shape. It is maintained by three types of bonds, namely ionic, hydrogen and disulfide bond. And hydrogen bond is much stronger than the disulfide bond. Ionic bond, the bond which is formed by the complete transfer of electrons between atoms or among atoms. And the hydrogen bond is formed when hydrogen involves in bonding with any other atom. And disulfide bond is formed when sulfur group or sulfide group involves in bonding with any other atom. For example, in aqueous environment, the most stable tertiary conformation is that in which hydrophobic amino acids are buried inside. Aqueous environment means the environment or the solution where the water or aqua acts as a solvent. Hydrophobic means water repellent, and hydrophobic amino acids means the part of amino acid which are water repellent. That's why this part are buried inside, while the hydrophilic amino acids are on the surface of the molecule, because here the solvent is water or aqueous environment, and the hydrophilic means water lover. So hydrophilic amino acids, that part of amino acids which are water attractor or water lover, and it is present on the surface of molecule. So in tertiary structure of protein, the protein will be in globular shape. Then the last is quaternary structure of protein. Quaternary structure is definitely much more complex protein and much more complex than the other three structures of proteins like primary structure, secondary structure and tertiary structure. In quaternary structure, polypeptide tertiary chains are aggregated and held together by hydrophobic interactions. Hydrophobic means water repellent, that part of the molecule which repel water from itself. Hydrogen and ionic bonds. These three types of bonds are present in quaternary structure. This specific arrangement is the quaternary structure. For example, hemoglobin, the oxygen carrying protein of red blood cells, exhibits such a structure. And hemoglobin composed of 574 total amino acids. As shown in the diagram, the primary structures lies flat or plane, the secondary structure coiled or in spiral form, and the tertiary structure double coiled or in double spiral form, and the quaternary structure much more complex than all these three types of structure of proteins. This is an important board question, so hope you will understand it. And do subscribe the channel, like and share the video, and give your feedback in the comment section below. See you in my next video. Till then, Allah Hafiz. Thanks for watching.